Good afternoon and welcome to the USDA Human Nutrition Research Center on Aging's weekly seminar series. Before introducing today's speaker, let me remind you to ask your questions for our speaker using the Q&A icon located on the webinar toolbar. And I would encourage you to ask your questions as they arise during the presentation. I take great pleasure today in introducing today's speaker, Dr. Sameera Talegawkar, who is an alumna of the Tufts University Friedman School of Nutrition Science and Policy. She performed her doctoral research and postdoctoral research with us here at the HNRCA. Sameera is currently an associate professor in the Department of Exercise and Nutrition Science and Epidemiology at the Milken Institute School of Public Health at George Washington University. She is a nutritional epidemiologist by training, and her research focuses on the role of dietary patterns and age-related changes in physical function. Other research interests include dietary assessment methodologies in diverse populations and the role of diet in chronic disease risk and underserved populations. Sameera is also the co-chair of the Jackson Heart Study Nutrition and Physical Activity Working Group and a member of the Scientific Council, and she is also a member of the 2025 Dietary Guidelines Advisory Committee. In today's seminar, Sameera will present some of her research related to dietary patterns, frailty, and physical function in two longitudinal cohorts of aging in a presentation titled "The Dietary Patterns and Age-Related Physical Function in Frailty." And with that, I will turn the microphone over to Dr. Talegawkar.
Thank you, Dr. Jakes, and thank you to the organizers for the opportunity to present at this week's HNRCA seminar series. As Dr. Jakes mentioned, I will be presenting some of my research related to dietary patterns and age-related changes in physical function and frailty. I will be presenting my work related to two cohorts, the Inkianti study of aging and the Baltimore longitudinal study of aging. This is a slide just talking about global aging demographics. The graph on the left demonstrates that the world population has, as it has grown and has currently reached about 8 billion people. But a huge change that has occurred is in the blue line that you see. There's a rapid shift in the demographics, wherein you see that those individuals aged 70 years and older have had a huge proportional increase. So while the population has also grown, the individuals who are 70 years and older have also had a significant increase. The graph on the right looks at a population pyramid, and as you can see, it is looking at decades from 1950 to about projections to 2100. And what you see over there is that these are countries, and the green bar actually represents a percentage of population in the countries less than 70 years and older. The white would represent a country which has a percentage of population 70 years and older, and then the orange is 20% or higher. And as you can see very clearly, there is along with the increase in longevity, you can see that there is a clear shift in the population demographics across the world, across countries. So it is not just certain the high income countries, but it's slowly moving towards a point where a significant proportion of the country's populations in the world, at least 20% or more, would be made up of older individuals. This kind of underscores and underlies the importance of studying older individuals. Of course, I'm speaking at the HNRCA, so I don't have to emphasize that. But what is important to emphasize is that as we look at or as we have increased or managed to increase the lifespan, one important consideration is actually improving the health span. So in this slide over here, it's just demonstrating the gap between what we call the health span and the lifespan. So as I've mentioned, we've increased life expectancy. Of course, it is probably due to changes related to healthcare, the advent of medications, etc. But what we are seeing is that while we have managed to increase the life expectancy, the number of years that an individual spends with some kind of a chronic disease or a disability is significantly high. So while we are living longer, we are living a significant portion of that life with a disability or a chronic disease, which therefore affects the way we function and perform in society. So the goal always of geriatrics and investigators in the field of gerontology is to compress that morbidity that happens towards the older ages. So the goal is always to kind of decrease this health span, lifespan gap to probably just before someone passes away, right? So we want to make sure that we are living longer lives, but the lives that we live are spent in a healthy manner, free from disability and chronic disease. Uh, which therefore brings me now to one of the kind of pressing concerns that generally occurs as individuals age. And this was, I think, first coined at Tufts by Dr. Irv Rosenberg, which is the condition of sarcopenia, which is the involuntary age associated loss of muscle mass, depth, and muscle strength. Which actually has this whole cascading kind of effect um, on physical function performance, then can have more detrimental associations and can lead to a condition known as frailty, which is more of a clinical condition, which then therefore can lead to a poor outcomes, including disability, a poor quality of life, falls, etc. Now, this is a very simplistic kind of way that I have represented, the whole kind of trajectory of physical function impairment, sarcopenia, and frailty. It is a lot more. These conditions can coexist at the same time, but it just makes it kind of easier to explain. The whole kind of trajectory from sarcopenia leading to frailty and then poor health outcomes. Now, the reason I bring up sarcopenia and frailty as well as physical function is because the research I'm presenting here today is related to frailty and physical function impairment. Now, unlike sarcopenia, which is to do more with muscle, frailty is actually a clinical condition. So you would probably see it in a geriatrician's office or sometimes in the observational studies that we have. But it is a state where there is increased vulnerability resulting from age associated decline in reserve and function. And it is not just related to muscle in this case, it is related to multiple physiological systems. And what happens with this is that the ability of older individuals to cope with any everyday stressors or acute infections or stressors is compromised. This in itself carries an increased risk for poor health outcomes, including falls, incident disability, hospitalization, and mortality. The kind of two schematics on this side talk about frailty onset and progression. So in addition to the fact about the biological kind of factors that might contribute to frailty, one has to always consider the demographic or the social determinants of health factors that can also lead to frailty. The slide on the bottom, or the figure at the bottom, actually is the classical kind of cycle of frailty definition that you might have seen in your readings when you've read about the Fried's frailty criteria. This is kind of more of the classical clinical frailty phenotype. You can basically say the starting point is frailty, which is the involuntary, as I mentioned, loss of muscle mass and muscle function. And then it leads with it a whole cascading effects where there is decreased strength and power, impaired balance leading to falls and injuries, decrease in walking speed leading to a decrease in activity levels of individuals who are frail, uh, leading to a decrease in total energy expenditure, and then basically chronic undernutrition. So the goal always of individuals working in the area of geriatrics and gerontology is to try and break the cycle of frailty. So frailty is a condition which in the early stages is kind of reversible. So the goal is always to try and see how can one prevent frailty and basically the downward decline that unfortunately invariably happens uh, once frailty has commenced and has not been allowed to kind of no intervention has taken place. Um, there are basically, well, there are actually several definitions of frailty. And this slide over here and the one I will speak about next actually talk about the two most commonly used definitions in frailty research. This particular definition is based on Linda Fried's frailty kind of phenotype. She put this forth, I think, about in 2002, I think, and it consists of like 5 factors. Weight loss, unintentional weight loss in the past year, weakness, which is assessed using grip strength, poor endurance, so a feeling of tiredness, slowness, which is measured using gait speed, and low physical activity, which can be measured using a questionnaire. So according to these criteria, if you test or if you report or if you show up with having 3 of the criteria, you are considered to be having frailty. And there is an intermediate or a pre-frail criteria, which includes if you have 1 or 2 of these criteria that are present. Um, in addition to the Fried's criteria, you will also come across what is known as the Rockwood Index or the Frailty Index. Now, unlike Fried's criteria, which basically are just 5, and if you meet them or no, it's you get a score of like a 0, 1, and it's counted. This is basically a count of deficits in health. Now, these deficits include symptoms, signs, disabilities, and diseases. So it could be things related to your activities of daily living, instrumental activities of daily living, chronic conditions that you might have, depression, etc. So, what is nice about this frailty index or this criteria is that it is not dependent on the having those for example, for the freeze, you need to have measured those 5 criteria. In this case, you can tailor your frailty index based on the data that is available to you in your study. So the reason I show both of these is because the research that I'm presenting actually has used both the freeze as well as the frailty index. Um, Now, this is another kind of a simplistic slide, I would say, but it makes it easier to explain that individuals, as they age. So, as you are middle-aged or you've entered middle age, you are generally independent. You are able to do your functional activities. You can do all your instrumental as well as your daily activities. So invariably, what ends up happening is that as you age and you get diagnosed with a chronic condition, and maybe you don't or you're unable for whatever reason to control that chronic condition, you start going down kind of a trajectory of a downward decline, wherein you might start showing dependencies or difficulties in performing independent activities of daily living. And then slowly you might slide into, as you get older and you are an older age, you might actually report difficulties in performing activities of daily living. At that point, when you report 1 or more concerns about activities of daily living, you are considered to have a disability and you might need actually help or assistance. Now, as I mentioned, this is a very kind of simplistic graph. What researchers generally would like to study is that how do we prevent a person from hitting that disability point? Because once you've hit that stage, unfortunately, you are going to need some assistance or help. So generally, what is measured or studied in observational studies is this difficulties in performing certain tasks, and the ones that are most commonly studied in of physical function or research include tasks such as gait speed or walking speed, or the assessment of physical function assessed using the short physical performance battery, also commonly abbreviated as SPPB. Now, this is basically a test that was developed almost 3 decades ago, and it is still used in research. It consists of 3 components: a time standing balance test, which gets kind of increasingly difficult with your feet side by side, semi tandem, and then tandem; a timed 4 or 5 meter test, depends on the researcher. And then timed multiple, in general, most often 5 chair stands, so standing and sitting up from a chair. Each of these tests are then scored and get a score from 0 to 4. There are certain cutoffs that are kind of put forth for an overall score of 12. Most studies use a 10 cutoff to indicate poor physical function. The reason I I am showing this test here and talking about it is because a lot of my actually results that I will be discussing are based on the short physical performance battery. Now, moving slightly from aging, frailty, sarcopenia, physical function assessment, I would like to talk about the exposure that I commonly have used in my research. I'm a nutritional epidemiologist, so I focus on diet, but I generally don't focus on a particular nutrient. I focus on dietary patterns. And I'm sure all of you are aware that we don't eat foods alone. We eat them in a combination. Recommendations also generally are made with respect to a dietary pattern. And in general, a dietary pattern approach is thought to capture the relationship between the overall diet and the interactions between food and nutrients as either health promoting or being health compromising much better. And hence, a lot of my research, most of my research has been focused on examining dietary patterns as an exposure variable. Now, this is also if you are students of nutrition or study nutrition, you know that you can derive dietary patterns using kind of two common approaches, an index-based approach or a data-driven approach. As you know, an index-based approach relies on scientific knowledge from previous investigations. So, some investigator has thought that a certain way of eating is healthful, and then generally they develop kind of guidelines or assign a score, on a way of eating. So, examples of this include the Mediterranean Diet Score, the Healthy Eating Index, the Alternative Healthy Eating Index. All of these are published, are available in literature, and have been used extensively in nutritional epi. The other approach is data driven. So it is basically examining either using cluster analysis, principal component analysis, or reduced rank regression, the patterns that emerge in your study population. Now, both are commonly used approaches. I have used both, but the research I'm presenting here today is related to the a priori or the index-based approach. So I am going to be looking at assessment or compliance with a pre-existing diet quality index or a current dietary guideline. And a reason I did that is because a lot of these indices and guidelines are it helps to have reproducibility across studies when you have something which is kind of scored, and there's a way of scoring it across different studies versus the data driven approach, wherein basically the study population or the study data kind of generates the patterns that are present in that particular population. While both of them are good, as I mentioned, I have used the index-based approach for the research that I will be showing here today. So when I started doing aging related research, obviously I needed a study or a longitudinal study that had diet and obviously aging related outcomes. And the first study that I kind of became aware of that had this information was the Inchianti study of aging. So the Inchianti study of aging is I think is basically based in Italy. It was funded through their Ministry of Health, but at some point it also was funded intramurally by the NIA. It is a population-based study of individuals living in the Chianti region of Italy. It has had basically several follow-up visits. So the baseline was conducted in 1998 and 2001, and since then it has had several follow-up visits. This is a study that is focused on aging. So hence it has really very good assessments of sarcopenia, frailty, grip strength, the SPPB, gait speed, etc. What is also nice about this study, being a nutritional epidemiologist, is that it has collected dietary data using a semi-quantitative food frequency questionnaire, which was validated in this study. So the FFQ that they have used is the the FFQ that was used in the EPIC cohort, the Italy site of that investigation. The study consists about 1,400 individuals. They range from 20 to 102 years. So they do have individuals who are in the younger decades of life, but the study is focused on aging. So the proportion of those who are younger than 65 years of age is much lower than those who are older than 65 years of age. Now, I just kind of wanted to give you a sense for like how did I come about with my whole focus on aging, dietary patterns, and where I am now. So the first investigation that I did in the Inchianti study was actually looking at the Mediterranean style diet, which made sense because this is a cohort based in Italy, and I thought that was kind of suited for that cohort. And physical function decline, which was assessed using SPPB. So this was a longitudinal analysis. I looked at a diet at baseline, and I examined adherence using the Mediterranean diet score, which was put forth, I think it was in 2002, and then looked at how how the adherence to the dietary pattern kind of resulted in SPPB or decline in SPPB. And what we basically found was that the closer you adhered to a Mediterranean style diet, the less likely you were to have like a less decline in SPPB. So you basically did better if you were with respect to physical functioning, if you were following a Mediterranean style diet. Now, since then, after that, I was interested in frailty. So I actually looked at Mediterranean style diet with frailty. And I found pretty much the same kind of story over there, where the higher the adherence to a Mediterranean style diet, you was associated basically with lower odds of developing frailty. This is longitudinal diet measured at baseline, and frailty measured over a follow-up period. So once I did this, got this published, one criticism that I received when I was, submitting this manuscript, and this has been several years ago that I submitted, was that you are measuring diet in older individuals, right? But we think, so this was a criticism given by the reviewers, that we think that diet during middle age and adulthood, so young middle age, is kind of more important than maybe diet in older ages. And that was a valid point. So I decided that I should consider examining diet in young adulthood and middle age with aging related outcomes. Now, unfortunately, the Inchianti study, as I mentioned, while it does have younger individuals, the proportion is much smaller. So I decided to actually pose this question in the Baltimore Longitudinal Study of Aging, or the BLSA. Now, the BLSA is actually an intramural cohort of the NIA, and it is one of the longest ongoing longitudinal studies of aging in the world. It has been active since 1958. It's what is known as an open cohort. So people kind of come in and exit the cohort at kind of different times. So unlike a closed cohort like in Chianti, where you assess baseline assessments and then you follow them over time, in this case, people can, depending on they can exit the cohort and then come back after 4 years and do assessments, or sometimes not come back. So it's a little bit of a different cohort, but it is really one of, as I mentioned, the longest running cohort of aging in the US. Now, what was another thing, what was is nice about the cohort is that it had diet measured. So diet was measured since I believe 1961 in the cohort, 1963 in the cohort, and this was done using 7-day diet records, which is kind of the gold standard, right? If you're ever doing a dietary assessment, that is a gold standard. So it was measured initially using 7-day diet records, and then in about 2008, it shifted to a food frequency questionnaire because keeping a 7-day diet record was rather burdensome, and hence they moved to a food frequency questionnaire. Participants basically come to the BLSA, so intramural to NIA, they come to the clinical examination site every 1 to 4 years, depending on their age. So if you are older, you would come a little bit more often than if you were younger. Women were not originally part of the cohort, but then joined BLSA in 1978, and currently there are about 1,300 individuals that are actively followed in the BLSA. Um, so this is a study that I did wherein I examined whether diet in young adulthood and middle age was associated with physical function, so SPPB kind of scores in older age. I'm just presenting the results. So this is not my main finding. It is, sorry, this is the main finding, but I just wanted to give you a background for what comes kind of next. Um, so what we did was we used latent class analysis to derive these trajectories of diet. And what we found was that in general, as people got older, they tended to eat a little bit better. So we ended up with two kind of dietary pattern trajectories. One was moderate and one was greatly improved. So moderate meant that if you were initially in a poor kind of diet quality kind of index, you moved to intermediate, and greatly improved meant you moved either from a poor to basically a much better. So no one actually deteriorated. Their diets kind of generally improved as they got older. And what we basically found was that um, even the slight improvement in your dietary pattern trajectory was associated with better physical function as you got older. So your diet was between the ages of 30 and 59, and the physical function we had was assessed in older age. And it was very clear that, what we saw for in Chianti, we pretty much saw similar effects wherein diet was associated. So better diet was associated with physical function as individuals aged. In this case, the index that we use, which I forget, I did not mention here, is yes, we use the alternative healthy eating index, because we thought that that was better suited, to check and examine adherence in a US population. So for example, in Chianti, we use the Mediterranean diet score. In BLSA, we use the alternative healthy eating index. Around the time that this work was published, there was a lot of research that was going on related to the Mediterranean DASH intervention neurodegenerative delay diet, or the MIND diet score. So there were a lot of observational studies that were coming out looking at the MIND diet with cognitive decline. And we had an opportunity to kind of examine the MIND diet with physical function decline, which we did. And we actually did find protective associations. So the more closer you adhere to the MIND diet, you were less likely to have physical function impairment as you got older. So this is kind of adding to the literature of dietary patterns with physical function decline or changes. So till now, we had basically looked at Mediterranean diet. We looked at the MIND diet. We looked at the alternative healthy eating index. We looked at all of these dietary patterns with physical function decline or frailty. Now, obviously a criticism again that we got was that observational studies, you really cannot talk about biological mechanisms with all of this information, which is absolutely right. So then we decided that we need to look at plasma metabolite markers of dietary patterns and frailty. So at the time of conducting this research, what we realized was that BLSA was actually conducting all of the assessments of the metabolome were taking place during that time. So this was a really good opportunity to see or observe what the biological effects of adherence to all of these different dietary patterns is. Given that we had the opportunity to look at it in the BLSA. So we actually proposed to do this as a grant application, but of course then we got criticized that we didn't have preliminary data. So hence we actually conducted a cross-sectional analysis to get some preliminary data to support our application. Um, and as you know, metabolomics is basically identification of diet potential biomarkers of diet quality using these high throughput metabolomics data that can isolate and identify thousands of molecules, thousands of specimens thousands of small molecules in biospecimens. Um, this approach is, kind of widely being used nowadays to look at the biological effects of adherence to dietary patterns. It's been done in population studies, and now more and more you are seeing results coming out from randomized controlled trials. At the time that we did these analyses, there was relatively less that had been done with respect to aging. A lot had been done with respect to chronic disease outcomes such as diabetes and heart disease. So we thought we should pose this question given that we already had derived several of the dietary patterns. The BLSA had assessed metabolomics, in one of their visits. So we thought this is a great opportunity to examine this research question. So that is exactly what we did. We looked at the plasma metabolite biomarkers of dietary patterns that we had previously demonstrated associations with age-related declines in physical function and frailty. And then we further examined the mediating effects of the diet-based metabolites on associations between the dietary patterns and frailty. In this case, we looked at kind of the three dietary patterns that we had already examined, which was the MIND, the Mediterranean diet, and the alternative healthy eating index. Now, if you remember what I had mentioned before, Mediterranean diet we had examined in Chianti, MIND diet we had already examined in BLSA, and same with the alternative healthy eating index. So we had some preliminary data to indicate that these patterns have been shown to be associated with frailty. And so we decided to query or examine these patterns in BLSA. Plasma metabolites were measured using the Biocrities life sciences kind of platform, and the appropriate kind of transformations, etc., were done to this data. I just wanted to actually thank my collaborator at NIA, Toshiko Tanaka, who's actually responsible for conducting a lot of these analyses for us. Uh, in this case, we looked at frailty using the frailty index, because as I mentioned, if you use the freeze criteria, you need to have like the 5 assessments at the same time. And that tends to be a little problematic in the BLSA because it's an open cohort. So you sometimes don't have all the assessments at the same time. So we decided to derive the frailty index using 44 variables, and the Rockwood criteria that were kind of present. So this analysis was cross-sectional. We looked at the adherence to the various dietary patterns and individual metabolites using linear regression models, with diet being the dependent variable and the metabolite as the independent variable. We had to correct for false discovery rate. Then we decided to select a set of metabolites that best predicts each diet score using elastic net regression. For the mediation analysis, we ran a model where the mediator or the metabolomic diet score was regressed on the diet score, and then frailty index was regressed on the diet score, adjusting for the metabolomic diet score. This was done to basically figure out how much of the association was mediated between the dietary pattern and frailty was mediated by the metabolomic diet score. These are some of our results. Panel A in this case shows the results of the elastic net regression analysis. Uh, what we found and what we decided to do was we selected 10, 41, and 59 metabolites using the elastic net regression for Mediterranean diet score, MIND, and alternative healthy eating index. There were 4 metabolites that were actually common to all 3 patterns, and 19 were associated with about 2, and 60 were unique to each of the pattern. Panel 2 is actually looking at the correlations between the metabolomic diet score and the observed diet score. And these correlations range from about 0.4 to about 0.56. Now, this is a similar range that has been seen in other studies which have looked at these correlations with respect to chronic disease-related outcomes. The metabolomic diet score explained about 23 and 31% of the variance in the observed diet score. So it varied, but it was between 23 and 31. When we did the mediation analysis, what we realized was that the metabolomic diet score mediated about 55 to 61% of the association between the MIND and the AHEI diet score with frailty index. Unfortunately, we did not find any association with the Mediterranean diet score in the BLSA. If you remember the Mediterranean diet score, we had found associations actually in Chianti, but not, and we had not looked at it actually in BLSA. But we did not actually find an association here. So in summary, what we found was that of the 466 metabolites that had been assessed, uh, there were a number of metabolites that were associated with the Mediterranean diet score, MIND diet score, as well as the alternative healthy eating index. Um, We also were able to identify a metabolic signature, and this included a wide range of lipid species, amino acids, their derivatives, bile acids, and vitamins. We did find that the metabolomic signature partially mediated the relationship between diet and frailty index, but only for the mind and the alternative healthy eating index. Now, the reason I don't discuss individual metabolites here is that there is always a criticism levied, and which is true, is that it is when you do these analyses, you always have to replicate it in another cohort, and this tends to be a little challenging because the platform that you use to do to do the metabolite kind of assays uh, kind of decides the metabolites that are that you're able to quantify. Um, so for our grant application, we decided to actually replicate it in the Health ABC study, since it is a cohort that actually uses the same platform as the Baltimore longitudinal study of aging. So as I mentioned, these analyses are all cross-sectional, but we should be hopefully getting funded for conducting the longitudinal and the replication analysis in the Health ABC study. Um, now, um, moving kind of slightly away from physical function decline, I, had the opportunity to conduct some analysis related to hearing in the NHANES. And what I found over there was that the Mediterranean diet score in the NHANES was associated so a better adherence was associated with better hearing status in older individuals. Now, the criticism, of course, of NHANES is that it's cross-sectional, and the fact that uh, you know um, as such, we had to combine several cycles of NHANES to actually get the audiometry data for the study. So we looked and saw that BLSA actually had dietary patterns measured, hearing measured using pure tone audiometry at some time point. So around 2012, they started measuring hearing status in the study participants. Um, Now, the reason I kind of pose this question is that hearing loss is a huge public health concern in older individuals, and is actually thought is one of the leading causes of disability. Not only does it, in addition to the fact that you are unable to hear, it can also lead to certain conditions of social isolation. So people might withdraw from society because they are unable to hear carefully. They are unable to hear. Um, and It can and has been shown to be associated with several other chronic diseases and functional decline. So there is a huge body of evidence that has linked hearing loss as individuals age with cognitive function decline in older ages. In the US, almost 63% of individuals aged 70 years and older report age-related hearing loss. So it's not a kind of a small concern. It's a rather huge burden in the US and worldwide. Um, Now, the previous literature, including ours, where we looked at NHANES, had shown kind of inconsistent results with limited number of longitudinal studies and less precisely measured hearing status or dietary intakes. Because since you need so kind of the gold standard is to do a pure tone audiometry assessment, and that is rather difficult to do if you have an observational study. So generally you end up with having a questionnaire related to hearing status or difficulties in hearing. So we decided, so this was like a side project, because I was just interested in hearing loss. I decided to measure the associations between the MIND diet, the Mediterranean style diet, and the alternative healthy eating index and the healthy eating index uh, using the pure tone audiometry among middle and older aged individuals. Now, a reason why I focused on middle and older aged individuals in BLSA is that it has been shown that even at younger ages, so about 45 years or so, individuals start demonstrating um, hearing loss. So it is not a condition that kind of starts or is commonly measured in older. It can start pretty early at around 45 years or so. Now, this is the consort diagram of as I mentioned, BLSA being an open core tends to be a little problematic. So it you don't have like a standard way of like you start out with so many people and everyone has the same assessments. As I mentioned, people kind of come in and out of the assessments. So in this case, we started with people who had the hearing evaluation done. We excluded individuals who had missing data, or had evaluations performed when they were less than 45 years of age. So we capped or started looking at hearing only at age 45 years and above. We also then obviously were restricted by those individuals who had diet data, and obviously valid diet data. So we ended up with a sample size of about 882 individuals for our analytical sample. As I mentioned, um, even though BLSA had started out with 7-day diet records, they then moved to a food frequency questionnaire because of the participant burden. So this data, as I mentioned, is post 2008, because the audiometry assessments were done starting 2012. So we actually used the FFQ data and derived all of the indices using those data. Audiometric assessment, as I mentioned, was done using the gold standard, and a whole range of hearing thresholds were examined. So you had speech level, low frequencies, as well as high frequencies. Um, High frequencies in general are the ones that start showing up as problematic even at younger ages. So if you're looking at the results, that's where you will, even at a younger age, start seeing changes to your hearing status. This is actually just a demonstration of how each of the patterns was scored. This is all published. Mind, Mediterranean, AHEI, and HEI all have published ways of scoring, and we stuck to the original scoring method. We used multivariable linear mixed effect models to examine associations between each of the dietary indices and hearing status. And obviously, we did model building. We also did a whole bunch of sensitivity analysis, because obviously the reviewers, and we know that exposure to firearm noise, work-related noise, etc., can affect hearing status. We also did adjustment for baseline hearing loss as well as any kind of missing data. So a whole bunch of sensitivity analysis was also performed. And this is, I agree, a very busy table, but all you have to look at is the fact that there were associations between most of the dietary patterns and hearing status. These associations were stronger at the higher frequencies, the ones that you end up losing earlier as you start aging. And in this case, just keep in mind, basically the lower score is better. So what we found was that a higher adherence to all of those helpful dietary patterns were associated with better hearing status as individuals kind of aged. Um, this is just summarizing the findings. What we found was higher scores, better adherence, better diet quality associated with lower pure tone audiometry, basically indicating better hearing status. We saw this across all, but it was actually much stronger for the higher frequencies in the better year. So just FYI, we examined both years, but we present the results only for the better year. We did find that there were limited associations again with the mind diet. And the reason we thought or speculated, this is pure speculation, is that the mind diet score doesn't have a PUFA component, which the others do. And that could be the reason that we were not able to capture any kind of a protective association of PUFA on the vasculature and the inflammatory processes involved in the disease process of hearing status or hearing loss as one ages. So I guess that's kind of summarizes my findings with, diet, I guess physical function, and then frailty. And then I thought I should present something new. This paper just came out last month in AJCN, looking at dietary patterns and hearing. I would like to acknowledge all of my collaborators and obviously the funding. My main collaborators at the NIA, Dr. Ferrucci, Dr. Tanaka, Dr. Simonsik, the Inkianti study PI, Dr. Bandinelli, um, Dr. Tucker, who actually leads the dietary assessment in the BLSA, collaborators, and my lab members at GW, Yichen, Tracy, and Jan. A lot of the assessments related to audiometry were conducted by Johns Hopkins. So investigators from there, and then a lot of the all of the work that was shown here was actually funded either by small grants, intramural, or by the NIA. So yeah, so thank you to the funding agency for funding this research. Happy to take questions. Thank you.
Thank you very much. Okay, and we do have a couple of questions already in the queue. So the first question comes from Larissa Constantino, and she asks, what may be the mechanism of the relationship between hearing and nutrition?
Oh, so it is thought that as I mentioned, the fact that a lot of hearing loss also occurs due to inflammation. So we know that diet or an anti-inflammatory diet or the higher fruit and vegetable intake is therefore protective. Another reason is there is a vascular mechanism. So any of the chronic diseases, including hearing, actually have a vascular basis. So anything that is protective against oxidative stress, damage to your blood vessels is actually going to be protective, therefore, of hearing. So maintaining your hearing status as you age. So those are kind of the mechanisms that are thought linking diet with hearing.
Thank you. The next question comes from Saidas, and first of all, she thanks you for joining us today, and then asks, with regards to the Mediterranean diet score not being aligned with the metabolomics in the BLSA compared to its performance in Chianti, how much of that is potentially due to the weak representation of the diet components in the usual US diet, I will add that, versus the score per se?
Yeah, no, I agree. So basically, Sai, you're asking that maybe using the Mediterranean diet score, which was meant for more of a Mediterranean population, for the US population was not a good idea. And I agree, probably was not. I should have used the US style Mediterranean diet score that has been set forth by the USDA. So I agree. It probably was the fact that our population was not very much aligned with the Mediterranean style dietary pattern score that we used. If we had used something that was more applicable to the US population, maybe we would have seen protective associations. But unfortunately, we have not derived that score in BLSA.
Next question comes from Michael Lusgarden, and he asks, hi, Sameera, you mentioned the difficulty in replicating metabolite associations across studies, but what were some of the metabolites that were identified as potential mediators?
Good. Oh, sorry. We had a whole list, and I apologize. Actually, don't have it in front of me. Y- Yeah, I'm sorry. It doesn't come like I cannot recollect all of them. We had several, and we talked about all of them in the paper. But yeah, this I have to get back to you. Like each of them, we did have them in supplementary tables in the paper. This has been published, as I mentioned. We were trying to get this as preliminary data for the application that we subsequently submitted. So we have this all documented, but offhand I cannot recollect. I cannot list them without error.
Okay, the next question comes from Daniel Promislow, and he asks, I really enjoyed your seminar. Do you
Thank
think
you.
that the metabolite associations are due primarily to metabolites that reflect dietary intake directly, or that diet mediates underlying physiological processes, and those in turn lead to differences in the metabolome?
That's a good question. If we get funded, maybe I can answer that using longitude. But yeah, no, that's a good point. Like there are so many other factors. I was like, this was all cross sectional, and you're absolutely right. It might be other processes and not really the metabolomic diet score, right? That you're talking. So yeah, I was not able to look at it in this. As I said, hopefully we get funded because it's made Payline, but we still wait. So maybe we can look at it, as I said, in longitudinal, because as far as I can remember, BLSA has repeated the metabolomic analysis again. I mean, the assay again. So we might have repeated data. So that might help in answering the question.
Okay, that's currently all the questions we have in the queue. I'll wait a few seconds here to see if any more pop up. But not seeing any more. Again, I want to thank you for a very interesting talk today. It's nice to see you back here at the HNRCA virtually.
Thank you, Dr. Jakes. Thank you, everyone.